Welcome to the deep dive. You know, we often talk about amazing tech, but sometimes nature just blows it away. Think about a dog's nose.
Oh, absolutely. It's staggeringly powerful.
Right. Far beyond what we humans can smell, even beyond our best machines sometimes. We know they sniff out drugs, explosives.
Standard stuff.
Important work. But what if that same nose could tell us something crucial about our health? That's what we're diving into today.
Yeah, the whole field of canine medical detection is fascinating.
We're looking at how dogs can detect human diseases just by scent, the breakthroughs, but also, the hurdles that are still there.
It's not straightforward.
No. And we're using a big review paper on canine olfaction, plus some really recent studies, especially focusing in on Parkinson's disease as a key example.
That Parkinson's work is particularly compelling.
So our mission today: let's unpack what makes these dogs such amazing detectors, figure out which diseases they can actually identify, and, maybe most importantly for you listening, what this could mean for diagnosing things much, much earlier.
Getting that early warning could be huge.
Okay, so let's start right there. Just how sensitive is a dog's nose? Can you give us something tangible? Because the numbers are just mind-boggling.
They really are. Try this. Imagine 2 Olympic-sized swimming pools filled with water.
Okay, got it. Big pools.
You dissolve 1 single teaspoon of sugar across both of them.
1 teaspoon in all that water.
Yeah. A dog could likely detect that. That's the kind of sensitivity we're dealing with. Parts per trillion level sometimes.
Wow, okay. That puts it into perspective. So biologically, what's going on there?
Well, dogs are what we call macrosmatics. It just means they have this extraordinary sense of smell and, crucially, olfactory memory, too.
So they remember smells really well.
Extremely well. And their anatomy is built for it. Inside that nose, there's a huge area of olfactory epithelium that's the scent-detecting tissue.
Much bigger than ours, I assume?
Oh, massively.
Yeah.
We have maybe 6 million olfactory receptor cells, dogs. They have somewhere between 200 and 300 million.
Whoa, okay.
Plus, they have that vomeronasal organ, for detecting pheromones. It all adds up to this incredibly complex system that can pick out tiny differences in molecules.
And how they sniff matters too, right? It's not just passive smelling.
Exactly. The way they sniff creates these specific airflow patterns. It directs the air, the odorants, right onto that epithelium.
Like optimizing the sample delivery.
Precisely. And each nostril samples slightly differently, helping them locate the source. It's remarkably efficient engineering by nature.
Which explains why they've been so useful for things like finding explosives or drugs for so long. That makes perfect sense now.
That groundwork really set the stage for exploring medical applications.
Right. So how does a disease even create a smell that a dog can detect? Where does that come from?
That's the core question, isn't it?
Yeah.
Basically, diseases cause metabolic changes in the body.
Changes in chemistry.
Yeah. And these changes release specific combinations of volatile organic compounds, VOCs. Think of them as tiny scent molecules released into breath, sweat, urine.
So each disease might have its own unique VOC signature.
That's the idea. The specific mix, the concentration, the type of VOCs, it varies. For viral infections, it's mostly the body's own cells reacting. For bacterial ones, it can be both the host cells and the bacteria producing VOCs.
Interesting. So let's talk about some specific diseases. The early work on non-infectious ones, like cancer, showed promise, but the results seem quite varied.
They do. If you look at cancer studies, bladder, prostate, lung, breast, others, you see sensitivity ranging hugely, maybe 19%, up to nearly 99%. Specificity, too, like 73% to 99%.
Why such a massive range?
Well, several reasons. Different cancer types probably have different scent strengths or profiles. The sample type matters. Urine, blood, breath, feces, they all carry different VOCs.
And the training, I imagine.
That's a big one.
Yeah.
There hasn't been a single standardized way to train these dogs across all studies, so you get variability. But even so, the potential is clearly there.
What about something like diabetes? Detecting low blood sugar, hypoglycemia?
Yeah, that's another key area. Dogs can learn to non-invasively alert to hypoglycemic episodes, hugely important for people managing diabetes.
How accurate are they?
Again, there's a range. Sensitivity reported between, say, 36% and 88%, specificity 49% to 98%. Compared to sticking your finger for a blood test, having a dog alert you is pretty appealing.
Definitely. And epilepsy, you mentioned that too. The idea they can sense a seizure coming.
That's the hypothesis, yeah, that there's a distinct body odor change before or during a seizure. One study using sweat samples was quite striking.
What did it find?
The dogs could tell the difference between sweat taken during a seizure versus between seizures with about 93% probability. And even more impressively, They warn the person before the seizure hit in 82% of cases.
Wow. Even if we don't know the exact molecule yet, that's potentially life-changing.
Absolutely. And interestingly, the results seem maybe a bit more consistent when we look at infectious diseases.
Okay, like what?
Well, pig malaria. There was a study, Guessed et al., in
Yeah.
2019, where dogs sniff socks worn by children.
Socks? Seriously?
Yep, nylon socks. And they could detect malaria even in kids who had no symptoms, with 72% sensitivity and 91% specificity.
That specificity is high, higher than the WHO threshold you mentioned.
That's right. It suggests dogs could be amazing for screening in high-risk areas, finding those asymptomatic carriers.
What about bacterial infections?
Also very promising, dogs trained on urine samples to detect urinary tract infections achieved almost 100% sensitivity and over 90% specificity for common bugs like E. coli and Staph aureus.
Incredible.
And C. diff, Clostridium difficile, that nasty hospital bug. They can detect that in stool samples with pretty good accuracy, too, like 78, 93% sensitivity.
And then there's the big one recently, COVID-19.
A huge amount of research popped up very quickly there. Dogs identifying SARS-CoV-2 infections?
Using what kind of sample?
All sorts. Saliva, sweat, urine, breath, masks, even clothes. What was really neat was seeing dogs trained on, say, saliva samples.
Yeah.
They could then identify positive sweat samples without specific retraining. It showed they weren't just memorizing one sample type, they were generalizing the actual scent signature of the infection.
So they understood the underlying COVID smell, regardless of the source.
Exactly. That generalization is key.
Yeah.
And also discrimination, being able to tell COVID apart from, say, the common cold or flu.
And the accuracy numbers for COVID?
Again, a range, but generally very high. Sensitivities from maybe 65% up to 100% in some studies, and specificities usually between 76% and 99%. Huge potential for rapid mass screening.
So, okay, this potential seems vast, but you mentioned Parkinson's disease as a really key recent breakthrough. Let's dive into that specifically. Why is it such a big deal for Parkinson's?
Well, the huge challenge with Parkinson's is catching it early. Diagnosis often happens years, maybe even decades, after the disease process actually starts in the brain. Symptoms are subtle at first.
Right, up to 20 years, you said. That's a massive delay.
It is. So the idea that there might be a detectable scent, an olfactory signature, for Parkinson's before those motor symptoms become obvious, that's the game changer.
And this signature comes from where?
It seems to be linked to changes in sebum, that oily stuff on our skin.
Yeah.
People with Parkinson's produce sebum with a different profile of those volatile organic compounds, the VOCs. It's driven by all the underlying changes, metabolic shifts, neurodegeneration, inflammation.
And this ties back to that amazing story about Joy Milne, the super smeller.
Exactly. It's a fantastic example of where this lead came from. She has hyperosmia, an incredibly sensitive sense of smell.
And she smelled something different on her husband.
Yeah, a specific musky odor. She noticed it 12 years before he was formally diagnosed with Parkinson's.
12 years? That's incredible.
It is. And initially it was anecdotal, but scientists took it seriously, confirmed it, and now she collaborates with research groups. It really kick-started this line of inquiry.
Wow. So that anecdotal clue led to proper scientific testing, like that double-blind study you mentioned?
Precisely. A really rigorous study involving the Medical Detection Dogs Charity, University of Bristol, University of Manchester. Real collaboration.
And the dogs. Bumper and Peanut?
That's them. A golden retriever and a black lab. They went through some serious training.
How long did that take?
Between 38 and 53 weeks. They worked with over 200 sebum samples from people with Parkinson's, including some who hadn't started medication yet, which is important.
And controls?
Yeah, healthy controls, but also people with other conditions like depression, anxiety, epilepsy, stroke, to make sure the dogs weren't just picking up on illness in general.
Okay, and the testing was properly blinded?
Absolutely, double-blind. Neither the dog handlers nor the researchers knew which sample was which during the tests. They even presented samples multiple times. Really robust.
And the results? How did Bumper and Peanut do?
Amazingly well. Sensitivity was up to 80% for one dog, 70% for the other. And specificity was even higher, up to 98% for one, 90% for the other.
98% specificity,
Yeah.
That's clinical level accuracy, isn't it?
It really is. On par with or even better than many existing diagnostic tools or other scent trials. And crucially, they could pick out the Parkinson samples even when those other neurological conditions were in the mix.
So they really learned the specific Parkinson's scent signature.
It strongly suggests they did. They weren't confused by other conditions.
Okay, so sebum seems key, but you also mentioned earwax. How does that fit in?
Right, that's another interesting angle. Earwax is actually mostly sebum, too.
Oh, okay.
But it might be a cleaner sample. It's more protected inside the ear canal, less likely to be contaminated by things in the environment, pollution, humidity, perfumes, whatever.
Makes sense. Less noise in the signal.
Potentially, yes.
Right.
Researchers Hao Dong, Dan Zhu, and their colleagues looked into this. They analyzed earwax VOCs.
And found specific markers.
They identified 4 specific VOCs, ethylbenzene, 4-ethyltoluene, pentanol, and another one, 2-pentadicyl 1-threol3-dioxylane, that seemed to be biomarkers for Parkinson's in earwax.
So instead of a dog, they're using chemistry to find the pattern.
Exactly. And they took it a step further. They developed an AI system, an AI-powered olfactory system, or AIO.
Trained on the earwax data.
Yep. And that AI system achieved 94% accuracy in distinguishing Parkinson's samples.
Wow. So that could be like an electronic nose for Parkinson's screening?
That's the potential, yeah.
Yeah.
A possible first-line, non-invasive screening tool based on those earwax biomarkers, mimicking the dog's ability with tech.
Okay, stepping back and looking at the big picture then, the advantages of using dogs seem pretty clear, right?
Oh, definitely. I mean,
Yeah.
non-invasiveness is huge. Speed, you get results almost instantly.
Cost-effective compared to big machines, probably.
Potentially, yes.
Yeah.
And they're mobile. You can take a dog almost anywhere, unlike lab equipment that needs specific conditions.
Good point. Safe, too, for everyone involved. Simple sample collection like sniffing socks or a skin swab.
And the ability to screen large numbers of people quickly. Think about airports or public events. Plus, training dogs for a new disease might be faster than developing a whole new tech test from scratch.
Okay, that all sounds incredibly promising, but clearly they're not in every doctor's office yet. So what are the real hurdles? The limitations?
Yeah, we need that balance. It's still early days for medical detection dogs, much newer than their use in security.
Right.
A major issue is standardization. Training methods, deployment, how results are interpreted. It's not uniform yet. Every dog is an individual.
Their performance can vary.
It can.
Yeah.
They're living beings.
Yeah.
They have good days and bad days. They need constant reinforcement training to stay sharp. It's not like calibrating a machine.
That makes sense.
Research findings can also be inconsistent sometimes, making it complex to draw firm conclusions across all studies. And it's crucial to remember, right now, this is seen as a screening tool, not a replacement for established diagnostic methods.
So it flags someone for more tests rather than giving the final diagnosis itself.
Exactly. Plus, training is intense. It takes almost a year, requires significant resources, and honestly, not every dog has the right temperament or focus for this kind of demanding scent work.
Finding the right canine candidates is tough.
It is. And even in that Parkinson study, while the results were great, we still don't know the exact VOCs the dogs were responding to. Why were some samples easier than others? And how does medication affect the scent? That study used drug-naive patients mostly. More research needed there.
So dogs might still beat an E-nose on pure sensitivity parts per trillion versus parts per billion, but the living creature aspect adds complexity.
That sums it up well. Incredible sensitivity, but biological variability.
Okay. So wrapping this up, the potential is just undeniable, isn't it? Especially for opening doors to much earlier diagnosis for diseases like Parkinson's.
Absolutely. It's incredibly exciting.
Yeah.
And even if the dogs themselves don't end up being the ones in the clinic routinely.
The data they provide is key.
Yes. It's invaluable for designing those electronic noses, those VOC-based tests we talked about, using the dog's nose to teach the technology.
So the dogs are like the pioneers showing us what to look for.
It's exactly leading us towards potentially rapid, cheap, non-invasive tools that can be scaled up. But we need more work. Standardization, reproducibility, getting official validation and certification. That's the path forward for this to become a mainstream screening approach.
It really leaves you thinking, though. If we keep following where these amazing noses lead, what other medical secrets might our four-legged friends help us uncover next?